Okay, we now want to talk about weed seed banks. Now, weed seed bank is actually the soil where most of the seeds will reside after shedding from the plants. From various studies, we now know that these weed seeds can remain for a long time. No wonder why we always get uh, weed problems. There are some uh, scientists or agriculturalists who wanted to take or who wanted to take this route of exhausting the weed seed bank so that we have a weed-free environment. This is quite a difficult task, and it may not be possible to create a weed-free environment which is permanent. So where are the weed seeds stored after formation and shedding? Most of the weed seeds are stored in the soil. No wonder why we have the term soil seed bank. Some could remain attached to the plant, and we can have aerial seed bank which is very small. So what is a seed bank? After weed seed shedding, seeds are deposited on the soil surface. This is where we find seeds on the soil surface, and in the soil form what is known as the seed bank. Seeds may also remain attached on the plant. This is the area seed bank, but this is not much. So we can have a variety of weed seeds on the soil surface. When the conditions become favorable, the seeds will actually germinate. And give rise to seedlings. Now this is shamba grass. At the base you can see, probably you can see the seeds which are scattered after shedding. Now the weed seed banks are near the soil surface. If you dig the soil deep down, you may not find any weed seeds. So here I've got a schematic representation of what can happen to weed seeds. Let us start with the green plant, that weed. If it flowers, it can produce seeds, and they are dropped to the ground as seed rain. Once they are in the soil, probably they will go into innate dormancy, induced dormancy, or enforced dormancy. They go in a state of dormancy and they are stored within the soil. Now, we also have weed seeds which are coming from elsewhere through dispersal. This is what we have been talking about. We spent a bit of time talking about the dispersal mechanisms which can bring in new stock of seeds into the fields coming from somewhere. Probably this looks like indirect investment. Then we have predation. Predation, you have got animals which can eat this seed. Some seeds may attempt to germinate, but they will fail. Some may be affected by pathogens. Some are at deep burial. And some are physically damaged. And some may actually become dead. That is the fate of these weed seeds. Probably this could be a better diagram where we focus on weed seed bank dynamics. Now, if you have got weed seeds which are in the soil, they are in two states. It's either they are non-dormant seed bank or a dormant seed bank. Non-dormant seed bank, you can easily withdraw if it was real money, in a bank, this is liquid money which you can easily withdraw. But some monies are fixed, dormant. You cannot just go there and withdraw them. So non-dormant seed bank can give rise to germination. After germination, some plants can actually die and not produce seed. But others which can produce seed through the seed rain or a dropping of the rain of the seeds, the seed can enter into the system. Or you can have a seed which is coming from far away via rain, wind, animals, machinery, seed dispersal, which we have actually covered, can contribute to the seed banks. Then you have decay. These are withdrawals of seeds from the seed bank. Pathogens, fungi, predation, you have got the birds, mice, and the insects. Why we refer it to as a bank? This actually mimics the actual monetary banks which we have. That's why we are using this sort of a model so that we can understand what is happening to the weed seeds as if they are everyday monetary transactions. So what is a weed seed bank? We have got the reserves of viable weed seeds present on the surface and in the soil. They consist of new seeds shed by the plant or older seeds in the soil for several years. So the seed bank is an indication of the past and the present weed populations. There are enormous numbers of weed seeds in the soil. In fact, it is the soil where the seeds are stored. So some weed seeds may eventually die within a few years, but others may persist for a long time. This is weed seed longevity. Excuse me, let me check something. We will continue. Let me check a certain message. Come in. Yeah. Yeah, yes, yes.
How are you today?
Yeah. Ndine lecture izvozvi.
Unoona lecture?
Izvozvi right now. Saka ndozopedza kuma 12.
Okay.
Yeah.
That's fine. Thank you.
All right.
Kutsvaga lunch. What do you do for lunch?
Yeah, after 12 ndapedza.
Oh, so I'm not
Eh?
What will you be having for lunch? Are you having lunch is two different things?
Uri kudzingwa nevanhu vakawanda. Yeah, sorry about that disturbance. So let's move on to talk about the density of weed seeds, which are linked to the cropping history of the land. Now, weed seeds may actually reflect what has been going on in a cropping field. So, seed density is actually influenced by the past farming and may vary from field to field. Thompson and Grime in 1979 divided the seed bank into two groups. The first group, they referred to it as the transient seeds, where you have grasses which are mostly dominant in the weed seed bank, which do not persist for more than 1 year. The second group is what is typical of the cropping fields, the persistent seed bank, where you have species of a wide range of habitats. These are the weeds in agricultural fields. Now, in grasslands, the majority of the seed bank is located within 2 centimeters of the soil surface, and nearly the entire seed population is located in the 15 centimeters of the soil. In conservation agriculture systems, the seed banks move closer to the soil surface. Seeds are in a better position to germinate and interfere with crop production. But under good weed management, the seed bank could be more easily reduced in conservation agriculture. Now let us look at the tillage effects on the weed seeds. Now tillage can affect the vertical distribution of seeds in the soil. In undisturbed conditions, the weed seeds remain near the soil surface. The example is the conservation agriculture or reduced tillage. And tillage with the moldboard plow buries the weed seeds deeper, probably to depths where they become dormant. And the reduced tillage can keep the weeds, the weed seeds close to the soil surface, like in conservation agriculture. This can reduce dormancy and encourage germination. And indeed, in some conservation agriculture systems, you find a lot of weed seed, a lot of weed infestation. The reason is that we have plenty of weed seeds near the soil surface, which can easily germinate. Here we have got examples of weed distribution in the soil profile under different tillage systems. Now, in moldboard plow, in conventional tillage, you get almost sometimes uniform distribution, or you get less weed seeds on the top, but you have plenty of them at the bottom. I am not going to talk about chisel plow, but what is common here is the moldboard plow and the reduced tillage, CA. In CA, it tends to be the opposite of conventional tillage, where you are going to have more weed seeds at the top and less as you go down. Now, there was a study which was done by Mavunganidze et al. In 2009. They found that the 0 to 10 centimeters depth had higher weed seed numbers than the 10 to 20 and the 20 to 30 soil depth levels. Probably it depends on the tillage which was being done in that field. It looked like reduced tillage. Now, fields which had been cultivated for a long time, more than 20 years, had larger seed banks than the recently opened fields. So the longer the field is under cropping, the more it is likely to have a lot of weed seeds in the soil compared to newly opened fields. So the soil acts as a medium of seed storage and growth for weedy plants. Now we want to look at the loss of seed from the soil. Now seeds are eaten by rodents and insects. That's how we lose the seed. or it could be through decay due to microbes. They may germinate and emerge. Certain experiments referred to as the Bureau experiments have indicated that there are some weed seeds which can last for 120 years and can still be viable, which are stored in the soil. This can indicate that weed problems are actually perpetual. It's something which we have to live with it. The Duvero experiments also indicated that some weed species could be viable up to 39 years. So seed longevity increased with all weed species, with all species, as the depth of burial is increased. So plowing action can bury weed seeds and they can be stored for a long time. And this seed longevity depends on the depth of seed burial, the amount and intensity of soil cultivation, crop management, and weed control programs can actually change or influence the weed seed bank size. Soil seed bank can be reduced by applying herbicides. Probably later on I will talk about how we can actually determine the weed seed bank size. Currently we have got 2 students who are trying to determine the weed seed bank sizes in agricultural systems or cropping systems in Murewa and Mutoko. Now, withdrawal of herbicides leads to increase of the seed bank to its original size within 1 year. So if we were using herbicides, we may manage to reduce the number of weed seeds in the soil. But the moment in which we remove the herbicides or stop using herbicides, the fields can revert back to their original status of high weed seed banks. How do the seeds enter into the soil seed bank? Or how are weed seeds deposited in the weed seed bank? The source of seeds, it could be weeds that are allowed to mature to produce seed. Weed seed production of standing weeds can contribute to the amount of weed seeds in the soil. Seeds can enter a field from outside sources, and the primary seed dispersal methods could be wind, water, animals, and humans, which we have already discussed. Surface irrigation water is also a potential to distribute weed seeds. Now, crop seeds which are contaminated by weed seeds. For example, wheat seed can be contaminated by Datura stramonium seeds in this country. Sorghum, pearl millet, and finger millet seeds are also contaminated with the weed seeds. And in fact, farmers do tend to share these crops. During the process, they end up, distributing weed seeds. Equipment movement can also distribute weed seeds. So many weed seeds can be eaten by animals and pass through the digestive tracts, but still remain viable. If we use manure, cattle manure, we end up distributing these seeds. Also, wildlife spread these seeds. So this is how they can come into the weed seed bank. Now, the germination and emergence can be a form of weed seed withdrawal, where some weed seeds are actually used during the process, but it can also tend to be an investment. Should the plants become mature, they will produce a lot of seeds. So seeds can also remain dormant. We've got seed dormancy, and we've got 3 types of dormancy. That is innate dormancy, induced dormancy, and enforced dormancy. Later on we'll talk about these terms. But I may mention now that innate dormancy is actually inherited. These are embryo status and the resistance of seed coverings to water and gas exchange. Induced dormancy is the germination does not occur even when the conditions are favorable. Enforced dormancy, these are environmental conditions, low temperature or lack of moisture, oxygen and light, which stops germination. Now let us look at the depth of barrier. And this can affect the germination and emergence. So the weed seed may remain stored in the soil. The ability of seeds to emerge from various depths depends on the soil and seed weight. Large seeds are able to emerge from greater depths, but we must also know that light penetration in the soil is affected by the silt, clay loam, and the sandy loam soil, and maybe light can penetrate up to 22 millimeters. So for those seeds which require light, they may be affected by the soil types. And the depth of barrier can actually affect light penetration. It is actually light which can trigger germination or not. So let us look at the factors which can affect seed bank size. The total number of seeds within the soil seed bank is determined by a number of factors which can include previous history of the land, that is weed control, weed species composition, weed population, tillage, frequency, dormancy characteristics of weed seeds, and lifespan, longevity. management of weed seed bank. How can we manage the seed bank? We can try to reduce the input of seeds into the seed bank via herbicide use, crop rotation, tillage, mulching, and also try to minimize weed seed dispersal. Probably this diagram may show all the factors which can affect the weed seed bank. So on our right, where we have this seed rain, we have got the seed bank, and we have got insects, fungi, and birds. We have also seed germination process. We have got dormancy, and we have got seed dispersal, bringing in the seeds. But on the other hand, we have got crop management. Things like mulch, fallow, intercropping, all of these human activities which can actually influence this root, this blue root. We can actually reduce seed deposits into the seed bank by these human activities. We can also actually we can increase seed deposits. Then we can also withdraw a lot of seeds because of these activities. But meanwhile, all of these processes are regulated by the agroecology, soil type, rainfall, and temperature. So these are the key drivers of weed density and diversity. Now I'm going to explain two methods of determining a soil seed bank. One method is popular, the other one is not. You will find out soon. The popular method of determining the weed seed bank is the weed seed germination method. What you do, you go into a field with an auger, which could be 5 centimeters in diameter, and take soil portions from 0 to 10, and put them in a bag. Take another 10 to 20, put in a separate bag. 20 to 30 in another separate bag. Now, after sampling the whole field, probably you need about 100 sampling points, which could be a lot of soil. However, increasing sampling will also increase precision, but there's a cost to it. How are you going to carry that soil? So the soil cores from the same depth are bulked together and passed through a sieve. The soils are then spread in asbestos trays. Then the trays are watered as necessary. What you simply do is to count weeds which are emerging, identify them, and record them. You pull them and remove them from the trays, and you have to wait until you get more weed seedling emergence. You can also disturb the soil after 1 month to encourage germination. You can do this for 3 months so that you have a reasonable number of you have reasonable numbers, or you can you can do it for a long time in order to exhaust the weed seeds. This method was done by Mamunganidze in 2009, determining weed seed banks in Mzarabani area in Zimbabwe. There are 2 students who are also using this method currently, working in Murewa and Mutoko, trying to understand the weed seed banks in those areas. This one is the difficult method, the second one, because there's a choice. This one is called the direct seed extraction method. So what you do after sampling your soils, the weed seeds have to be separated from the soil using what is known as the flotation or sieving methods. This can be a messy process. Soil samples could be soaked in solution of sodium bicarbonate or sodium hexametaphosphate, sodium hexametaphosphate or sodium bicarbonate for a short time. You need a jet of water to remove clay and silt particles. Once seeds have been isolated, they can be identified. So you should be able to identify the weed seeds to their species level, which can also be difficult. Viability tests will also be done using the tetrazolium tests to determine whether the seeds are alive or dead. Probably you can have a look at this paper which determined the weed seed bank in a maize field. In maize fields. Anyway, these are the data from that paper. You can also use these references where these studies were actually done. Other papers as well on seed banks, and this is it on soil seed banks. Stop streaming.